Okay, hi, I'm Kelly Ruggles, and I'm from NYU Medical Center, and I want to welcome you all to the Multi-Omics Integration and Application session. Thanks for all being here. Today we're going to go through 6 talks that are going to cover a large and diverse range of data types, including genomics, transcriptomics, proteomics, metabolomics, and the microbiome. And these talks are going to cover both the development of new tools to integrate and analyze these types of data, and also ways we can apply these methods to push biological discovery. So we're going to start with with Pratik Jagtab, and he's going to tell us about his work in Metaproteomics for the Future, democratizing functional analysis of microbiomes via community-based informatics development and dissemination.
Good afternoon, everybody. On behalf of the Galaxy P team and the multiple authors on this project that we have done, I'd like to thank the ASMS organizers for giving us the opportunity to talk about some of the work that we have been doing in the field of metaproteomics. The Galaxy P team from University of Minnesota has been working in the field of multi-omics, especially proteogenomics and metaproteomics for the last 5 years, and has developed multiple tools and workflows to enable research in these fields. Today I'm going to talk about metaproteomics, so using basically a community effort to address some of the questions in this emerging field of metaproteomics, which is study of proteins that are expressed by microorganisms. So as I said, this was a collaborative effort involving researchers from all across the world, from various institutes. These included opinion leaders, researchers, developers, as well as users. So just to give an idea about metaproteomics. So metaproteomics is basically a tool that one uses to address questions in the field of microbiome research. So some of you might have actually heard about microbiome research. So microbiome research is basically a study of microbial gene expression in response to any environment. And this could be applicable to clinical research, or this could be also applicable to environmental research. Most of the research has been focused on correlating taxonomy of microorganisms to various physiological conditions. And there have been some really good papers in the last few years correlating microbiome to physiological conditions such as diabetes, atherosclerosis, mental health condition, arthritis, and so on and so forth. There's also been a lot of researchers who have been involved in who have worked in environmental microbiology and have used metaproteomics or microbiome research as a way to look at how microorganisms react to environment. Most of the research in microbiome has been focused on using DNA molecules. So metagenomics is an extremely advanced field. If you give a search on NCBI PubMed, you'll see a lot of publications in this particular field. Necessarily what is done is DNA from samples, either clinical or environmental samples, are collected and then are subjected to 16S rRNA studies so that one can perform taxonomy analysis on that, or whole genome shotgun analysis, also called as metagenomic shotgun sequencing. That also helps you to understand the composition of your microbiome. And eventually, multiple studies have actually been done to correlate the taxonomy to a particular physiological condition. What we are interested, and for the reasons that I'll explain later, is study of metaproteomics. So using metaproteomics as a way to understand how a microbiome reacts to the environment. And the advantage that one has with metaproteomics, which is a study of proteins that are expressed by microorganisms, is that obviously use proteins, use mass spectrometry to identify peptides and proteins, is that one identifies the function of a microbiome. So if you want to compare 2 states of, let's say, disease state and normal state, you should be able to actually identify proteins that are expressed by these microorganisms and then decipher the functions of each of these states. We believe that this actually helps you to come closer to the mechanistic understanding understanding of how a microbiome reacts with the environment. And one more thing I'd like to mention is metagenomics has a really big community of researchers. In fact, there's a national microbiome initiative involving mostly metagenomics researchers, while metaproteomics is still an emerging field. And I think we as a community need to start getting integrated into the large microbiome research field. So, metaproteomics, why is it a challenge? Metaproteomics has been a field that has been in existence for the last 12 years, while single organism proteomics has been there for much more than that. One of the big differences in single organism proteomics and metaproteomics is the search databases that I use. I mean, there are multiple challenges, but this is one of the main challenges. The search database for single organism proteomics is basically a result of the genome that is sequenced for that organism. It is converted into a protein FASTA file, and that's what is used for your research. Most of the times, these search databases are of small size. The complexity is not very large. There's only a single organism, maybe with the contaminant sequences that one puts in. While the metaproteomic databases are much larger because you are looking at multiple organisms, and sometimes you are looking at public repositories, which are much, much larger. The database also has multiple homologous proteins. As a result of that, there have been challenges that have been put forth, which are more pronounced for metaproteomics. One of them is that search algorithms need to be developed so as to accommodate ability to search large databases. Protein grouping becomes a challenge, not only within the single organism, but also across multiple organisms. Identification statistics take a hit because of its effect on false discovery rate analysis. Generally, your sensitivity of identifications goes down. Generally, when you identify unique peptides from your metaproteomic data sets, you can use it for taxonomy analysis. While if you identify peptides which could be unique or shared, you could go back and find out which proteins they come from, and hence find out what is the function of these proteins. Now, for all of this, you actually require software tools, and you also require various processing tools that are required. And not all of these tools run on a single environment. You need to have some tools work on different environments. So in order to kind of address this, we presented last year. We presented our solution of using Galaxy, which is a web-based interface, as a bioinformatics platform. Now, Galaxy is a web-based interface that facilitates software accessibility to web bench researchers. So basically, anybody who is not a bioinformatics expert can still use some of the tools and workflows that are available in Galaxy. It ensures reproducibility, so you can use these workflows for multiple replicates. You can assimilate multiple software and put them into workflows, and then that actually helps you to run this analysis in a seamless manner. We developed some workflows for metaproteomic analysis, and we published this in 2015. And we use these workflows for multiple research studies, including one research that was published in 2015 on dental plaque analysis. And we also have some two other papers, one on bronchoalveolar lavage fluid metaproteome, and another one on cervical vaginal fluid, which is in works. So we realized that when we use these workflows, we actually had a limited number of tools or limited number of resources that we could go to. For example, for database generation, we used public database repositories. There are a lot more options available. For database search, we used the SciAx software protein palette. For functional analysis, we generated outputs which could be analyzed through Megan. And for taxonomy analysis, we used inputs for Megan as well as another tool called as UniPep. So we wanted to extend this repertoire of tools and use them in workflows. In order to do that, we reached out to some researchers or metaproteomics experts all around the world, and also developers who would help us in identifying as well as installing these tools in Galaxy environment. And we generated a user list. We discussed this over emails, and we prioritized these tools and workflows and decided a kind of a date for what we call as contribution fest. So the difference between a contribution fest and email exchange is that you kind of decide on 1 or 2 days wherein all these developers and users meet online on a chat group or on GitHub, and then discuss about details which could be missed via email communication. So as a result of this, we could extend our list of tools that we are interested in from the limited number that we have to the ones that have been shown here. And then we started working on this contribution fest. So in December 2006, led by a Galaxy developer named Bjorn Gröning, we started packaging and testing of tools on Galaxy. We generated GitHub issues. Tools were developed and packaged. They were tested with Conda and added to the Galaxy toolshed. So Galaxy toolshed is basically a repository of tools that is available so that researchers or developers can download these tools and use them on their own Galaxy instance. So these tools are now available for worldwide Galaxy users. So there was a lot more details and a lot more discussion that went on. And if you want to know details or even a summary of these of this contribution fest, you can go to this link and get more information on that. Now, as a result of this, we could, as you could see, we could expand the list of our software tools. The ones that are shown in dark are the ones that we could implement, and the ones that shown in light blue are the ones that we are still testing or developing. And hopefully, in the next few months, we should be able to have at least some of them in Galaxy. So as a result of having all these tools, we could put these tools into workflows and make them work. In fact, we used some ocean data sets, which were published in 2016 from a lab in University of Washington, and used those data sets to demonstrate that these workflows can be used right from database generation to functional analysis. So one of the things that was enabled because of this was you could actually take shotgun metagenomic databases and convert them into, which are FASTQ in FASTQ format, and convert them into a protein FASTA file with peptides. And this was as a result of a software named SigSkill, which was developed from Nunn Lab at the University of Washington. And at the other end, for functional analysis, we could extend the capabilities of Unipept to not only perform now taxonomy, but also perform functional analysis on smaller data sets, as a result of which now, I mean, again, this was because of the help and work that was done by the Unipept developers, and this kind of helps us to get a slightly more extended pipeline that we had earlier. As I said earlier, we presented this as a workshop at the annual meeting of ABRF, the Association for Biomolecular Research Facilities, in March this year. You can go to the link below if you want to know more details and documentation of this particular workshop. We actually went a step further and also generated or have a Galaxy instance now wherein researchers can go and access this instance to look at the inputs, outputs, as well as workflows and documentation for the workshop that I mentioned earlier. This instance is on Jetstream, which is a cloud instance that Indiana University provides, and we have a collaboration with them. Those who are interested in seeking this out, please go to the Jetstream website, and you should be able to get an idea about how you can access not only this instance, but also any other instances that you might need for your research. So as I said, there's documentation, tools, and workflows available. You can go to the website shown below if you want to test these workflows. Now, looking into the future, obviously we have an extended set of lists. There is a lot more that can be done. There have been quite a few papers that have been published which have shown the importance of databases, using the right database so that you can come as close to your metaproteomic analysis as possible. Also, there have been other papers by opinion leaders in this field which take a much more forward-looking approach and what next in metaproteomics. So just to summarize some of these interesting papers as well as thoughts, There is a lot that one can learn from metagenomic analysis. There is a large community of researchers, there is a large community of statisticians, developers, and also rich visualization tools that they have developed. So it will be nice to see if we could integrate these into metaproteomics studies as well. Quantification is, as much as it's done in single organism proteomics, is not practiced as much in metaproteomics. So it will be nice to follow up with the work from researchers who perform, let's say, data independent acquisition or any other methods. Multiomics, there is a lot of researchers who have started doing metatranscriptomic analysis and also metabolomic analysis in microbiome systems. It will be nice to integrate that to bring a lot more systems biology view or analysis to the system under study. In the various discussions that I have had with researchers, quality control of not only the quality of data that's generated, but the quality of spectra that are reported is also something that needs to be discussed about and addressed. There are quite a few groups who have been working in the field of targeted proteomics, metaproteomics. So it will be nice to learn from them. In fact, there's a group at the Woods Hole Oceanographic Institute who have been doing targeted metaproteomics on the ocean, at the ocean level. So, and they're also putting up ocean metaproteomics portal. So there is a lot of community level involvement that's taking place. I put up this slide just to say that, wish we have versatile analytical workflows that not only can address the microbiome of the surfer shown here, but also the ocean metaproteomic, ocean that's underlying it. We have a vast amount of data, and hopefully with these workflows will help us sail through it. Lastly, I would like to basically mention that Galaxy P team has had multiple posters. So we had posters on using Jetstream as an instance for both proteogenomics and metaproteomics. There was a poster on using multi-stage method as well as a sectioning database method on Tuesday. There was another poster on using visualization capabilities within Galaxy after database search is done. And also today there was a poster on metabolomics by Art, and basically there are workflows now available for even metabolomic analysis. If you are interested to know more about Galaxy, please come to the workshop today in room 135, 136. It's a hands-on workshop, not only on metaproteomic analysis, but also proteogenomics and Galaxy in general. This is a hands-on workshop, so please bring in your laptops, and hopefully we'll be happy to take you through some of the workflows that we have built. Lastly, I'd like to thank multiple researchers, including Professor Tim Griffin, who's the PI on the Galaxy P grant. We also work very closely with Minnesota Supercomputing Institute. Thanks to James Johnson, who's the senior developer at the institute and helps us package most of the tools there, and multiple researchers and developers around the world. Thank you.
Thanks, Pradeep. So there's a question from online. How can you resolve the issue of overlapping databases from multiple species and quantifying, specifically the possibility that a unique peptide in one species may not be unique in another?
Right. I mean, that's a very valid question. I think at least the way we address that in our analysis is we merge the databases and basically avoid any duplication of any of the protein sequences. But in terms of analysis, you can quantify these peptides, and if you have unique peptides, you can use them to assign it for taxonomy. And if not, then you can use it for assignment to function. So I don't think you lose information there. Quantitative information actually would be useful either way. You can say this taxonomy species went up based on a unique peptide, but if not, if you can assign it to a protein, you can always say this is the function that was elevated, or.
Great.
Yeah.
Thanks. And I have one question. Are you guys integrating a specific pipeline for metatranscriptomics analysis to go sort of in parallel with this pipeline?
Not right now, but we would be interested to
explore
that.
Great.
Yeah.
All right. Thanks. Join me in thanking him again for a wonderful talk.